Hey everybody, this is Eric with FlightBridgeED and welcome back. I wanted to do a two-part podcast on cellular respiration. I'm real excited about this topic, and over the past few years I've started studying this topic as far as glycolysis, the Krebs cycle, how cellular respiration really affects our patients. I may be a little bit of a nerd in that way, but I really have found that we really can affect a lot of change when we optimize oxygenation. So I've tried to figure out how to do that. I know that when we first come into this business, whether we're an EMT or a nurse, we learn good BLS procedures. We learn how to manage our patients with a BVM and how, we want to put patients on a non-rebreather and give them 100% oxygen. But I think when we further our career, we kind of lose sight of that. I know I did. I think if you're honest and you're sitting here and you probably reflect back, we get excited about the new advanced procedures we can do, intubation, ventilator management, etc. And if we really think about the pathophysiology involved with cellular respiration, and if we always revert back to, on most presentations, patient presentations, if we revert back to how can we optimize oxygenation and how is this optimization going to affect overall patient morbidity, mortality? Is it going to increase it or is it going to decrease it? And I think you can argue it's always going to decrease mortality most of the time if you do this aggressively. So I wanted to take a little bit of time and do a few podcasts on cellular respiration. All right, so we're going to start with glycolysis. I'm not going to go through a master's degree level introduction on glycolysis or the Krebs cycle or electron transport chain. I've tried to break this down to the simplest form. I am going to go into somewhat in depth in some areas, but hopefully it'll be easily understood. I know in FPC, CFRN review classes that I've taught, that this isn't hit as in depth as I'll probably hit this here. So if you're preparing for those exams, this might be a little bit better to kind of get an overall understanding of how all this works. So this whole process has three phases. So we have glycolysis, we have Krebs cycle, and we have the electron transport chain. All of those things combined produce ATP, and we know that ATP is essential for cellular function. It's essential to produce heat, and for overall homeostasis. All right, glycolysis can happen with or without oxygen, and as far as this lecture goes, I'm going to go primarily in depth regarding aerobic metabolism, which means with oxygen. I will hit really quick on anaerobic, explain the difference there, but just remember anaerobic is without oxygen and aerobic is with oxygen, but glycolysis can happen with or without. We're going to again talk about with. So glycolysis is referring to the breakdown of sugar and the breakdown of oxygen to produce ATP. All right. So if there's oxygen available, we're going to utilize the Krebs cycle. We're going to utilize the electron transport chain to produce ATP. If there was no oxygen available and we were in an anaerobic state, we would bypass the Krebs cycle, we would bypass the electron transport chain, and the pyruvate molecules that are broke off from the glucose molecule would essentially produce lactic acid. All right, and that's going to be a whole other podcast on lactate, lactic acidosis, and how that kind of affects our patient. But for purposes of this, we'll just strictly talk about aerobic metabolism. So let's explain kind of how all this works. So we got a glucose molecule, and the glucose molecule, remember, is essential. We have to have lots of sugar in our body for energy. Our body, though, doesn't need, if we have a low sugar state, our body can produce or generate sugar off of our fatty tissues, off of our proteins. All right, so we have an abundance of sugar, and we have mechanisms in place to produce sugar if needed. All right, so our glucose molecule is made up of 6 carbon molecules. All right, so 6 carbon molecules are attached to that glucose molecule. So just kind of picture a big old fat glucose molecule, and you have 6 little branches off of that molecule, and those are all carbon. All right, so our whole body is made up of carbon, and we know that carbon is a very powerful energy source. All right, so when I start talking about oxidation, and oxidation is losing electrons, basically what we're doing through this oxidative process is we're breaking down each one of those carbon molecules attached to the sugar or attached to the pyruvate. And we're utilizing this. So this process keeps happening over and over and over to break down each sugar molecule. Okay? So remember that, because it's going to be important here in a few minutes. So at this point, we have, again, 6 carbon molecules attached to our glucose, and we need to start this whole glycolysis process. All right? So we have oxygen as well. So we know we have a aerobic state. And from this glucose molecule, we're going to get that broke down into 2 pyruvate molecules. All right? So we started with 1 sugar molecule with 6 carbon, and now we have 2 separate pyruvate molecules broke off from that glucose molecule. All right? So this is the beginning of that conversion. Each one of these pyruvate molecules has 3 carbons attached to it. So if you kind of go back, we had 6 carbons attached to the 1 glucose. Now we have 3 carbons for each pyruvate. So 3 plus 3 is 6. That's where we get the 6 or the 6 carbon molecules on the pyruvate. All right. From this point, we start the chemical oxidation process using the Krebs cycle. So the Krebs cycle basically is the whole breakdown of these electrons, this losing the electrons. And it really is a short process. The glycolysis aspect of the breaking out of the sugar into the pyruvate is a very simple process. From that process of the glycolysis, we produce 2 ATP molecules. Okay, that's going to be important for later. Through the Krebs cycle, again, it's a very short process. We're beginning that oxidation process. We're beginning the process of breaking down those carbon and losing those electrons. From there, we're going to start the process of utilizing the electron transport chain to produce energy. All right? So, glycolysis produces 2 ATP. Krebs cycle produces 2 ATP. So right now, based on that, we have 4 ATP kind of just sitting out there that's produced. Again, every time this happens, we lose one of those carbon molecules off of that glucose, or off of that pyruvate, I should say. All right, so now we're in the electron transport chain process. So we have the 2 pyruvate molecules, and those pyruvate molecules now need to be converted to something else. And this is kind of where it gets kind of complex. I'm not going to go into in-depth definitions. I'm going to throw out some different names here. And again, my understanding of a lot of this is pretty general. It's not, I would say, even, I would probably struggle in some high-level, college courses, to be honest with you. I know I would. So at this point, we have our 2 pyruvate molecules, and they need to be broken down into something else. All right? So through the Krebs cycle, through glycolysis, we have 4 ATP molecules. We have the 2 pyruvate molecules, and those pyruvate molecules are now broken down based on the oxidation of those carbon so each carbon, right? So we have 6 carbon. So each carbon on that pyruvate molecule is going to produce 10 NADH and 2 FADHs. All right? Why is that important? Well, to kind of sum up this really quickly, each of the 10 NADHs produce 30 ATP. All right? So basically, one NADH molecule produces 3 ATP molecules. For every 2 FADHs, you produce 4 ATP molecules. All right, so essentially, for every FADH molecule, you produce 2 ATP. All right, so based on the NADH breakdown and the FADH breakdown, just on those, utilizing the electron transport chain in an aerobic state, we produce 34 ATP molecules from 1 carbon off of that pyruvate. All right, so remember, we're breaking down, we're losing electrons. We're trying to get rid of those carbon. Carbon is an essential, huge energy force. All right, so we have 4 ATP molecule that have been, or ATPs that have been produced from glycolysis in the Krebs cycle, and through the breakdown of the NADHs and the FADHs in the electron transport chain, that produces 34 additional ATP molecules. All right, so how does it do that? Well, this process is still being figured out. There's many scientists out there that are studying this and doing tests on this, but it's thought to happen using what's called an ATP synthesis engine. All right, and we've utilized our sugar. Right, we started off with a sugar molecule, and we said that we have to have oxygen. We need oxygen for this as well, for this chemical reaction. Well, this is where that oxygen comes in. All right, so basically we're using oxygen, and we're going from a high energy source to a lower energy source, and we're breaking down that oxygen molecule to run or pump this ATP synthesis engine. All right. So the oxidation and the breakdown of this oxygen molecule and the other chemical processes causes a process called hydrolysis. And we've heard of hydrolysis. And this hydrolysis produces H2O. H2O is, again, what helps drive that synthesis engine to produce ATP. All right, so let's recap that. So you have your 10 NADHs and you have your 2 FADHs for every carbon molecule off of your pyruvate, right? So your pyruvate is broke down, essentially, each pyruvate molecule is broke down 3 separate times. Each time it's broke down, we get 10 NADHs and 2 FADHs, which produce 34 ATP molecules. How those NADHs and FADHs produce that ATP is through this process of breaking down the oxygen molecule into water, called hydrolysis, and that hydrolysis turns that synthesis pump. So kind of think about, these hydroelectric dams, the water that's turning and producing electricity. It's kind of a similar process. We're breaking down our oxygen, we're producing it to H2O, through this chemical process, and I think there's a step there that I've missed to do this, but basically we're gaining a hydrogen ion off of this NADH is what we're doing, and we're adding that to the oxygen that's giving you the H2O. All right, so we have hydrolysis that produces this high energy state, and it produces and runs this synthesis engine that produces the ATP. All right, so this process, for every time this happens, just think about this going in a circle. It produces 34 ATP molecules. We had 2 ATP molecules produced from the glycolysis. We had 2 ATP molecules produced through the Krebs cycle, and that gives us 38 ATP molecules per chemical reaction or per process. You may have seen literature, books, textbooks, or been taught that for every time this happens in an aerobic state, you produce 36 ATP molecules, and that is correct. I know I just stated 38, but remember, every time this happens, it takes energy to do this. So every time you do this, the cell or this chemical process utilizes 2 ATP molecules to produce this chemical reaction. So you have to minus that from the 38. And so you do get 38, but what's actually utilized and what's there at the end is going to be 36 ATP molecules. So to kind of recap, through the glycolysis we have 2 ATP, through the Krebs cycle we produce 2 ATP, and through the electron transport chain and the breakdown and the utilization of that ATP synthesis engine and the utilization of oxygen, the breakdown of oxygen in water, we gain 34 ATP from that. All right, so that's kind of the simplest way I can explain that. Hopefully that makes a little bit of sense. So at this point, let's start talking about oxygenation. I wanted to start with glycolysis and kind of how our body breaks down things and produces energy at the cellular level. Remember, everything happens at the cellular level, but now we need to talk about how oxygen is introduced to our body and what it means to us. All right, so remember, in order for us to survive, we have to extract oxygen from the atmosphere, and we have to transport that to our cells to be utilized for metabolic processes, right? Some cells can produce energy without oxygen, and that's what's called an anaerobic state or an anaerobic metabolism. But remember, that's a very insufficient way of doing things. Your body or the mechanisms that do utilize this can only do this for a very short period of time. In contrast, when we talk about organs that really utilize or need oxygen, like the brain and the heart, they need a continual supply of oxygen to maintain homeostasis or for perfusion. All right. So if we have a lack of oxygen or anoxia, the brain and the heart are the most sensitive or the most affected. So, obviously, when we talk about a CPR situation, a cardiac arrest situation, the big emphasis now is good deep compressions for a prolonged period of time. Essentially, you're never stopping. You're doing fast, deep compressions, allowing full recoil, and it's thought that keeping that map pressure higher, that we do have good oxygen-rich blood that can be circulated for a few minutes. We don't necessarily need to be giving ventilations for a few minutes. That the negative pressure in our chest, the recoil when we let that chest recoil, is going to draw on some oxygen. So that's really important to understand that our brain and our heart need this. It needs that continual supply. So how does all this happen? Well, we need to understand what diffusion is. And diffusion is basically going from a higher concentration to a lower concentration. We learned this in biology class or chemistry class when we were in ninth grade. And so how does it relate to us? Well, we think about oxygen diffuses from our alveoli to the capillaries, and basically you're getting an equalized pressure. So your alveoli to the capillaries until the PO2 in the capillary is equal to that of the alveoli. All right, so if you can kind of think about that, you have two different pressure gradients, and those two pressure gradients are equalizing, and that's how the oxygen is diffused. All right, the process is normally completed by the time the blood has passed about 1/3 of the way through the pulmonary capillary. All right, so this is a very quick process in a normal lung. It's very rapid. In order to make this happen, obviously, though, we have to have everything needs to be perfect, and so when we start talking about these patients that have a shunt or they have a ventilatory perfusion mismatch, obviously that can alter this, and we'll talk about that here in a few minutes. All right, so how is this all produced or transported to the tissues? Well, oxygen is transported from the air that we breathe to each of our cells in our body, and it's done through that pressure gradient that I said. We go from a higher concentration to a lower concentration. And through this process, we take air in, it goes through, it's circulated into our alveoli, it's diffused into our capillaries, gas exchange occurs. So the alveoli to the blood. How are we going to get the oxygen molecules from the alveolus to the blood? Well, blood returning to the heart from the tissues has a low PO2. So remember, our PO2 range in a normal healthy adult is 80 to 100. Well, when we have blood returning from the heart, so it's deoxygenated blood, our PO2 level is about 40 millimeters of mercury. So that's still above what our oxygen-rich environment is, our 21% oxygen-rich environment. And so at this point, it travels to the lungs via the pulmonary arteries. Remember, pulmonary arteries are the only arteries that carry deoxygenated blood. We know that arteries always carry oxygenated blood, and so this is the one that doesn't do that. The pulmonary arteries form pulmonary capillaries, which surround the alveoli. All right, so oxygen diffuses, moves through the membrane separating the air and the blood, and that's the alveolar membrane, and through the high pressure in the alveoli, which is about 100 millimeters of mercury, to the area of low pressure in the blood in the pulmonary capillaries, which is 40 millimeters of mercury. So remember, like I said before, you have a low pressure gradient. Your pressure is going to go from higher to lower. It's trying to equalize. All right, the blood returning from the heart has a PO2 of 40. Our blood moving out, going into our tissues to perfuse, is about 100, and so they're trying to equalize. All right. After oxygenated blood moves into the pulmonary veins, which returns to the left side of the heart to be pumped to the systemic tissues. In a perfect lung, the PO2 of the pulmonary venous system or blood would be equal to the PO2 in the alveoli. All right, so let's talk about this. 3 factors that can cause PO2 in the pulmonary veins to be less than your PO2. Well, that's where we start talking about a ventilator perfusion mismatch, ventilation perfusion mismatch, a shunt, or slow diffusion. All right, so let's talk about ventilation perfusion mismatch. In a perfect lung, you've got to remember all alveoli would receive an equal share of alveolar ventilation. So basically, ventilation and perfusion would be equally matched. You're going to get both sides is going to be equally ventilated. Both sides is going to get equally perfused. When you have somebody that's got maybe decreased lung fields, diseased lungs, you may have a very marked mismatch between ventilation and perfusion. All right, some alveoli are relatively overventilated in some cases, and others are relatively overperfused. So what does that mean? Well, basically, you can think about it in the most extreme form. When we talk about a shunt, where the blood flows past the alveoli and no gas exchange is occurring, and because there's a shunt in that alveoli, it's not allowing that oxygen-rich blood to diffuse into the alveoli. When we talk about a well-ventilated alveoli, where we have a very high PO2 in the capillary blood, it cannot make up for the oxygen not transferred in the underventilated alveoli with a low PO2 in the capillary blood. All right. Even normal lungs have some degree of ventilator perfusion mismatch. The upper zones are relatively overventilated, all right, while the lower zones are relatively overperfused and underventilated. And that's where you'll kind of hear me talk about functional residual capacity and how that applies, about capturing as much as we can as far as PEEP, and really doing a good job in recruitment. We do recruitment through our inspiratory phase, our volume, whether it be volume or pressure, but our inspiratory phase is going to recruit alveoli. Our PEEP is going to maintain that recruitment. So all that to be said, that's very important in this process. And so if you remember that your upper zones, obviously, that are easily reached, are going to be overventilated, and your lower zones, the lung fields and the bases and such, are going to be overperfused and underventilated. So we got to kind of find the middle line there. We got to find the spot where we have an equal share of ventilation and equal share of perfusion. So let's talk about a shunt, where we have this ventilation perfusion mismatch. All right, so a shunt occurs when deoxygenated venous blood from the body passes unventilated past the alveoli and enters the pulmonary veins. So, like I said before, you have all this oxygen-rich blood that passes by the alveoli, but there's a shunt there, and you don't have any gas exchange. All right. So why would this happen? Well, if you had atelectasis trauma, which is essentially alveolar collapse, you have consolidation of the lung, you have a pulmonary effusion, pulmonary edema, if you had edema in the small airways, etc. We could go on and on and on. ARDS. So how do we decrease the detrimental effects of a shunt? Well, we need to fix the ventilator perfusion mismatch and optimize the oxygenation. So we can do that in lots of different ways. Again, whether you go with a pressure-driven mode and you're trying to increase that functional residual capacity, you're trying to recruit more alveoli, you're trying to ventilate those lung fields that aren't getting ventilated and try to equalize that ventilator perfusion mismatch. Like we said before, we want that to be equal. All right, so some bad things can happen though because of this. Our body has mechanisms to try to correct itself, and so one thing that can happen based on this ventilator perfusion mismatch is you can have your blood vessels in your lungs try to adapt to this, and they vasoconstrict, and so this causes reduced blood flow to the areas which are underventilated. So those lower lung fields that are underventilated, our lung recognizes that, it tries to vasoconstrict, it tries to adapt, and this basically causes something called hypoxic pulmonary vasoconstriction, and it reduces the effects of the shunt, but over a long period of time this can damage the lungs. This can be this is going to be a bad thing. So, we need to try to correct for this. So how is oxygen carried in the blood? Well, we know that oxygen is carried in the blood by hemoglobin. It's carried in two different forms, but mainly the hemoglobin, and then there's a small amount that's actually dissolved in the plasma. All right, and we got to remember that. It's just a very small amount. Each gram of hemoglobin, I think, carries about 1.3, I think, or 1.4 mils of oxygen when it's fully saturated. And so it's really important to understand that globin can only hold so much. To be fully saturated, it can hold just so much. So you can't overdo it. So when we start talking about FiO2 and giving an abundance of oxygen, sometimes that's not going to make a difference. So what does that mean? Well, for every hemoglobin that's got 1.3 mils or 1.4 mils of oxygen attached, how does that translate to something that we can understand? Well, we know that a normal hemoglobin level is about 15, approximately. We'll just throw that out there. And so if you have a patient that has a normal hemoglobin level of 15 and all of their hemoglobin molecules are saturated with 1.3 or 1.4 mils of oxygen, that's going to kind of translate to a PO2 of 100, all right? And so basically what that is 1.3 mils of oxygen. When you look at the whole concentration of hemoglobin, is going to give you 200 mils of oxygen total in your body that your hemoglobin is holding on to be dispersed to the tissues when everything's perfect, right? So you have to have a good hemoglobin level, right? We can't be anemic. You've got to have, no shunt. All those things have to be working perfectly. So let's recap that. So every gram of hemoglobin carries about 1.3, we'll say, mils of oxygen when fully saturated. Every liter of blood that we have has a hemoglobin concentration of 15. That's going to equal to approximately 200 mils of oxygen when fully saturated. That our hemoglobin has oxygen molecules attached to it. At this point, only 3 mils of oxygen will dissolve in every liter of plasma, right? So like I said before, there's very little oxygen that dissolves in the plasma. If the PO2 of oxygen in arterial blood is increased significantly by breathing 100% oxygen, let's say, you just still get a very small amount of oxygen that is dissolved in the plasma. Remember, we can't put any more on the hemoglobin, so it's got to go somewhere. So, we disperse that out and we get rid of that, and we have a small amount that's dissolved in the plasma. With that, though, again, you're not going to have any normal significant increase in the amount of carried by the hemoglobin because we already know we have a fully saturated patient, probably greater than 95%. And you're not going to have any changes there. So we need to consider the adequacy of oxygen delivery to the tissues. And so there's 3 factors that need to be taken into account. All right, so we got our hemoglobin concentration, what we talked about. That's your oxygen carrying capacity, our cardiac output, and again, our oxygenation. All those combined is going to make things good, right? And really, we're going to talk here at the end about the Fick principle, and the Fick principle is essentially that. It's how your body and your tissues utilize oxygen and drive cardiac output, essentially. All right. At this point, we're going to talk about oxygen stores. So, our body obviously stores oxygen, and in spite of the great importance of oxygen, the stores of oxygen in our body are small. And really we would be unable to sustain life for a few minutes if we didn't have it. Remember, if breathing ceases, oxygen stores are limited. They're going to be shunted. They're going to be utilized for our heart and our brain. And the amount of oxygen in the blood depends on the blood volume, and the hemoglobin concentration. So when we talk about our patients that are very sick or septic, you got to think about how all these little things factor into cause significant deterioration in our patients, and that's where we start getting to end stage organ failure and such. So really, we have to think of a lot of different ways, and I think this is why I became so intrigued in this. Is when you are just doing standard street medic medicine, or doing, a little bit of ER medicine as a nurse or whatever, you don't think of these things until you get into critical care. You start getting into the real sick ICU patients, and you start understanding all these different factors and how it relates to our patients. Obviously, the blood volume is essential. We have to try to monitor that. We need to know what our hemoglobin concentration is, and if it's low, we need to correct that. All right. Well, how can we correct for the oxygenation concentration? Well, like I said before, we have something called FRC, and that's functional residual capacity, and we're trying to fill those dependent lung fields in the bases, the ones that we talked about that are underventilated and maybe overperfused. And so we need to try to equal all those things out. We need to open up the lungs. And so in a normal adult, your FRC, or your functional residual capacity, is about 3 liters. All right? And that's at the end of the expiration process. So we want to try to reach as many little alveoli sacs as we can. All right? While breathing air, the total stores are small, and because the major component of these stores are oxygen-bound hemoglobin, only a part of these stores can be released without an unacceptable reduction in PO2. So what that's saying is that, although we have, like we said before, we have the 200 mils of oxygen bound to our hemoglobin. If we have a normal blood volume, our hemoglobin is normal. Remember, a hemoglobin molecule carries about 1.3 mils of oxygen, and if you do the math, that's going to give you about 200 mils. Well, that's all fine and dandy, but really that's not a lot. So only a small part of these stores can be released without an acceptable reduction in PO2, and if you did a blood gas, that's where you're going to significantly see that. To kind of give you an idea how this is if you have some type of shunt or some type of problem, if you took that same hemoglobin molecule and it was only 50% saturated, so what would that would be, like 0.65 mils of oxygen per hemoglobin, you would have a PO2 of 26 millimeters of mercury. So that's horrible. And if you can see if that's done for a long period of time, how that's going to affect you. If you go back to the beginning of this presentation and you start talking about glycolysis and you talk about aerobic metabolism, well, this is where you get that anaerobic metabolism state, and you start getting that lactic acidosis, where we have the pyruvate molecules. They can't convert. They can't break down those carbon chains, and they can't produce energy through the NADHs and the FADHs because it doesn't have the oxygen. Remember, you have to have that oxygen molecule to drive that ATP synthesis pump. Well, if you don't have that, you have this anaerobic state. You can't do that. You bypass the Krebs cycle, you bypass the electron transport chain, and it produces lactic acidosis. Again, we're going to talk about lactic acidosis or lactate levels in another presentation, but just know that for the purposes of this presentation, lactate levels in your body initially were thought to be a marker of tissue perfusion. Although that may be somewhat correct, it's now more looked at as a mortality morbidity marker, biological marker. And so basically a lactate level is 2.5 or below is normal. Anything above that, for every point above 2.5, you have a 10% mortality rate. So it's kind of looked at more so in a sick patient and how they're doing and how they could do. So we've talked about all these things. So how is this going to affect our patient, right? And when we talk about oxygenating our patients, and we did this whole presentation, and the beginning of this, I started talking about how can we optimize oxygenation, how can we do things right based on how we learned in our initial careers, providing good oxygenation. Well, if we kind of take what I just said regarding the hemoglobin, if hemoglobin is only 50% saturated in a sick patient, and that gives you only a PO2 of 26, and how that can cause all these cascade of problems, we need to think about pre-oxygenation. And we do this, let's say, in an RSI patient. All right, so let's talk about that real quick. When we breathe 100% oxygen, it causes a large increase in the total stores. All right, so remember, we haven't had complete, all that oxygen hasn't dispersed everywhere. The major component of this store is now in the lungs, and 80% of this oxygen can be used without any reduction in hemoglobin saturation. So your PaO2 is still about 100. Remember, we have that equalization. We have 100 in our alveoli, and we have 40 millimeters of mercury in our pulmonary capillaries that's deoxygenated. And so we still have that PaO2 of 100 millimeters of mercury. Well, in a pre-oxygenated state like this is a good thing. So this is the reason why pre-oxygenation is so effective. We haven't utilized all those stores, and that's why it's so important to increase that functional residual capacity to make sure we optimize our inspiratory phase. If you haven't listened to the ventilator lecture yet, remember we don't want very high tidal volumes. That's a bad thing. That leads to that cascade, and that causes ARDS. We don't want ventilator-induced lung injury, and so high tidal volumes do that. But we want a tidal volume that's going to be, physiologic. And our tidal volumes that we take in are 400 to 600, mils of air per breath. So that's where we need to stay at. We don't need any more than that. And if you take into account that we have a patient that's got a sick lung, on top of that, we definitely don't want to overinflate. So we want to recruit based on a good inspiratory pressure or volume, and we want to maintain that recruitment via PEEP. All right, so just remember that. And by doing that, we're going to optimize that pre-oxygenation. We're going to use that effectively. All right, so I said in the beginning of the presentation, or in the middle, that we have something called the Fick principle, and the Fick principle is, again, something that's kind of still being studied, or it's very complicated. And the Fick principle is essentially an equation that describes mathematically the amount of oxygen that we can deliver to the body and the volume of oxygen that is then consumed by the body. It relates back in part to the exercise physiology and how blood and oxygen function in relation to one another. So essentially, cardiac output equals oxygen consumption plus arterial and venous O2 differences. And this drives your cardiac output. So, if you're one of the employees that I have as an educator below me, and I've commented on your charts, and I've talked about optimizing oxygenation and how it affects cardiac output, that's essentially what I'm saying. This is a very important concept. So we think about it in simple terms. Basically, oxygenation consumption and uptake directly affects cardiac output positively and negatively. And as far as oxygen physiology, that's all I have for this podcast. I know it's very in-depth. If you have any questions or comments, or there was something that I said that you just don't understand, please email me at eric@FlightBridgeED.com, and I'll definitely get back to you. I may put this on a blog, and I appreciate you listening. Thank you.